Okay, hi everyone, my name's Maricia, and today I'm going to be talking about my graduate project, which looks at the effect of ROS generation at the oxic-anoxic interface in wetlands. So as I said, my name's Maricia, I'm a PhD student. I work with Dr. Brandy Toner and Dr. Bill Arnold in the Civil and Environmental Engineering Department. And I, as Mason said, I study chemistry, so I would consider myself a soil and water chemist. So I wanted to start off today by talking a little bit about the places that I work. And when I was deciding to come to graduate school, I really was interested in studying field work. And so my two field projects are at the Spruce site, which is located in northern Minnesota. And then my second field site is the Prairie Pothole region, which is located about 30 minutes outside of Jamestown, North Dakota. But I would say that I spend a majority of my time in the laboratory, and that could be processing samples, running reactive oxygen species experiments, or prepping samples for beam time. So let's see. This kind of cuts off the beam time photo. So this is a picture of me and Caroline Pierce, who's another graduate student at the Canadian Light Source, running samples for Zane's. Well, I was helping with her project, but that was for Zane's spectroscopy. Okay, so I study wetlands, as I said. The first is the Prairie Pothole region. They are outlined in orange. And then the other is the Spruce site, which is outlined in green. And as you can kind of tell from the photographs with the gloves on, the soils are pretty different. And I wanted to highlight that by kind of going over some of the site characteristics and why I'm interested in studying them. So the Prairie Pothole region has high sulfate reduction rates, it has high methane emissions and high carbon dioxide emissions. And kind of the wetland characteristics of it are these small, shallow ponds that are depressional and discharge wetlands, and they're seasonal. So the water level is very dependent on the time of the year. They receive water from precipitation, regional groundwater, and runoff. And they were formed from glaciers. So the parent material is glacier till. It has hummocky topography with a relief of 33 meters. And because of that, the chemical characteristics are that there's quartz, feldspar, dolomite, calcite, pyrite, and high concentrations of sulfur, iron, and organic carbon. Now, if you compare that to the Spruce site is an ambitrophic peatland, so it is a carbon sink and stores a lot of organic carbon in the soil. It receives rainfall only through precipitation, and its parent material is organic, primarily coming from sphagnum moss. It also has hummock and hollow topography, but the relief is much smaller, 10 to 30 centimeters. And as I said, the chemical characteristics are that it's nutrient poor with high organic carbon presence. So these two field sites are both wetlands, but they have very different characteristics within them. Okay, so moving on, I study a specific reaction called the dark Fenton reaction, and it looks something like this. So it happens between reduced iron, oxygen, and it produces oxidized iron and reactive oxygen species. I like to call these sequential redox reactions. And I studied them at the oxic-anoxic interface of wetlands, which is likely to be very redox active. And the component that I'm specifically interested in studying is the hydroxyl radical, which is an unselective oxidant of organic carbon, and it reacts extremely quickly. So as fast as it's being produced, it's going to want to react with that organic carbon in the soil or in the sediment. And I just wanted to make a note that there are Fenton-like reactions. These can happen with iron minerals like iron clays, iron 3 oxyhydroxides, can happen with complexed iron, dissolved iron, and sulfides and reduced dissolved organic matter. So in the Prairie Pothole, the reason why I'm interested in studying this reaction is, as I said, they're semi-permanent wetlands, so they have saturated soils. There's likely anoxic conditions that produce reduced species like iron 2 and sulfides. And then they also have oxygenated surface waters and frequent mixings, so likely interactions happening between oxygen and reduced species. And then at the Spruce site, while I'm not necessarily running these reactive oxygen species experiments, the reason why these are important, could be important for that field site, is that there's saturated soil present in the cattle tongue, oxygenated soil in the acryltome and iron presence. And another just kind of interesting thing about reactive oxygen species generation is that it's poorly understood and studied in soils. Okay, so the last part of my presentation is going to go over the experimental design of my reactive oxygen species experiments. So there are a couple of key requirements for this. The first is that there is no exposure to oxygen happening before the reaction is starting. The second is that the reaction needs to proceed in the dark. And the third is that oxygen needs to enter the enclosure. So I have a vessel on here. It's wrapped in tinfoil to prevent any photooxidation. And then I have this box that I cover over the reaction to basically prevent any light from entering in. And it has the slits at the top to allow oxygen to enter. And the reason why I have to cover it is because iron also can be photooxidized. And so just to prevent any photooxidation and because I want to study them in the dark. So the way that I do this is I prepare a solution in the glove bag, I take it out of the glove bag, and I expose it to oxygen. And then I basically take time samples while I'm running the reaction and measure it on the HPLC. And what I'm interested in tracking is hydroxyl radical generation. And I'm able to do this in this reaction because I use a fluorogenic probe, which basically just means that I add in this compound called TPA. It reacts with hydroxyl radical that's happening in the vessel, and then it fluoresces. And so it's easily tracked on the HPLC. And from that, I'll calculate a rate and see how fast it's being produced. And that's it. Are there any questions?
How far along are you?
So I have done some reactions with iron ferrous sulfate, and those didn't go so well. So now I'm going to try running my my sediment samples that I collected, so dried sediments, and I'll be doing that this month.
Right. So what do you do in the Canadian light source?
Yeah, so the question was, what do I do at the Canadian Light Source? So at the Canadian Light Source, what I was specifically doing during that trip was helping Caroline Pierce run some sulfur-zanes. But I was just recently at the Advanced Photon Source, which is in Chicago. And what I studied there is zanes and XF spectroscopy, which helps me understand the iron speciation of the sediment in soils. Yes.
I got a question for you.
Yes.
What's your, I didn't get your hypothesis. After you're done with your experiment, what are you going to find?
Yeah, so I'm interested in tracking the generation of hydroxyl radicals. Hydroxyl radicals are likely going to be reacting with organic carbon that's present in the soil sediment, and that could be a potential reason why there's such high carbon dioxide and methane fluxes in the prairie potholes.
Okay, thank you.
Yeah.
So this is, do I understand right, that this is a potentially kind of unrecognized or underappreciated source of hydroxyl radicals in these systems?
Yes, yes. And I also want to say I study the abiotic side of it, but the biotic interactions are still going to be happening in this. And so they're also important. I'm just specifically studying how hydroxyl radicals could be formed through an abiotic process that could be kind of speeding up along these biotic processes that are happening. The hydroxyl radicals are unselective oxidants, which means that they're going to react with like organic carbon that's present in the sediment or in the pore water and the surface water. And so they're going to be basically breaking down this organic carbon into something that becomes more readily available for microbes to use in their respiratory processes. Yes. Yes.
What are the particles where hydroxyl radical
Yeah, so well, they can be, I think they're important in a lot of different places, but specifically they've mostly been studied in aquatic environments. But I think that they could be important in wetlands because there is saturated soil and also oxygenation that happens in these.
Any more questions?
Yeah, I have a question.
There we go. Go ahead.
All right. Is this a reversible reaction? Can it go the opposite direction?
I'm not sure if it's reversible, but I do know that, like, for example, iron, because it is redox active, it's able to be oxidized or be reduced through like cyclical processes. So what could happen is, like, let's say some of this iron is oxidized, it produces hydroxyl radical. Well, then that iron is still present in like the sediment or in the water, and so it can actually be re-reduced and go through this process again.
Thank you, Rachel.
Awesome, thank you.